Hello everybody, in this video we're going to talk about the cell cycle, 4.2, Molecular Genetics, Lecture 2, the cell cycle. Today we're just going to talk about how cells divide. As you can see over here, we got one cell and then we got two cells, right? And we've done this in some of our classes on Tuesday where we start with one cell. It got to grow, right? It grows and grows and grows, and then it snips right in the middle, and we got two daughter cells. We unfortunately don't call them son cells, they're called daughter cells. But this process, right, as you already know, the cell have to grow first before it divides. Otherwise, every time your cells divide, it's going to get smaller and smaller and smaller. Then you're going to have nothing left, right? You can't have one skin cell bigger than another skin cell. That's not going to work out too well. So today we're going to talk about how this whole process happens. A kind of an overview. It's not, super detailed, but basically what you need to know about the cell cycle. Here we go. There are all these words that we're going to talk about. It looks really scary, but we'll get through it. It's going to be fine. But take a look at it, and then at some point when you're reviewing for the quiz or for the test, come back to this and then take another look. We got some cell division. So one question that I want you to think about right now is, what is it? Why do cells do it? Why is it important to me? So please pause the video and answer these questions on your paper if you're watching this video for class right now. All right. So to me, right, cell division is not this kind of division, is cell dividing, right? But why would cells be dividing? Sometimes unicellular organisms might use cell division literally as is reproduction process, right? We go from one cell to two cells. Now we go from the first generation to the second generation. And that's great, right? But for us, multicellular organisms, we use cell division to help us grow, right? When you go from this tall to this tall, you obviously need more cells to allow that to happen. We also use cells to repair, right? So for example, if you break a bone, unfortunately, or if you break anything, you break your skin, right? Your cells will make up for that breakage, which is not a word, but your cells will divide and kind of fill in the gaps, right? So for growth and repairs, for multicellular organisms, having cell division is very important, right? Also, just staying alive, we need a lot of skin cells. We need a lot of red blood cells. It's pretty important for us. Why is it important to me? Yeah, that's important enough. But if you have other ideas, feel free to write them down. All right, now what I need you to do is to pause the video and then draw this entire thing, okay? So you can use a water bottle, you can use a dog treat jar, whatever you want to use, but your goal is to draw up a pretty decent circle. If you don't have anything circular, that's okay. If you can't trace the bottom of your bottle, that is fine. You won't fail this class by not drawing a perfect circle, but go ahead and draw this circle, and you need to divide it into 4 pizza pieces, but you need 1 giant piece, 2 semi-normal piece, and 1 pretty normal pizza piece. So 4 pieces, okay? Once you're done drawing that, you want to label these 4 parts. So basically what happens is, right, we go from our 1 cell to 2 cells. We go through 4 big phases. The first phase is called G1, which is also called the gap 1 phase. So go ahead and write this down. We have the S phase, which is called the synthesis phase. So what are we synthesizing? Really, we're synthesizing DNA. So it's called the S phase. But really what happens during this phase is DNA will replicate, right? So each strand of your linear DNA as a human being, I hope you're a human. If you're a monkey watching this, I'd be a little concerned, but no judgment over here. But anyway, even for monkeys and dogs, whatever, all of us eukaryotes, we have linear DNAs, and all our linear DNAs will go from this one line into an X shape that we learned about back on Tuesday, right? But really, it's more like this. This is 1 copy, and there's another copy of the DNA, and it goes into an X shape after it's synthesized and condensed. You can't actually see the S phase, the X shapes over here yet, but they're there. And then we have we'll talk about that some other days. G2, we have the gap 2 phase. So this is another gap, right? We're not quite there yet. We're almost there. We're getting there. That's okay. And then lastly, we have the M phase, which is where the actual event of cell division take place. So M in this case stands for mitotic. So this is our mitotic phase. And there are two parts to it. The first part is called mitosis. So you might have learned about this, or at least heard about it in middle school. Mitosis is how our skin cells, for example, go from 1 cell to 2 cells to 4 cells, blah, blah, blah. Mitosis is our cell division process, right? And then, but there's another step called the cytokinesis. In AP Bio, we go cytokinesis. We might have done this freshman year for you guys last year. But cytokinesis is the actual division of the cytoplasm and the cell membrane, going from one cell, the actual pinching and allowing one big cell to divide into two cells. So that's cytokinesis. So the M phase has two parts to it. There's mitosis and there's cytokinesis. You might also notice that there are these red lines. These are called checkpoints. So we're going to talk about that. These are checkpoints. You can kind of think of them as, TSA at the airport, where you can think about coming to Georgetown Prep if you come on campus sometimes, right? There are these regulations that we have to go through. We need to do our QR code. We need to check for temperature. You get your COVID test. So that's basically the idea, right? Before our cell divides, we have to make sure that our cell is worth dividing. We're not making more and more of the damaged cell. That would not be good for our body, right? If we have a cell that already has DNA that's very damaged and doesn't contain all the genetic information, or even worse, it might become cancerous. You don't want more of those cells, right? So your body will, your cell, really, one cell, every single one of them, will go through these checkpoints to make sure that things are all right. We actually should be dividing. Should we be dividing? Okay, we should be dividing, okay? All right, am I okay? Is my DNA fine? Okay, now we're dividing. Okay, so that's the idea. We'll talk about each one of these checkpoints and exactly what goes on over there. The last thing that you want to notice over here is we have G1, S, and G2 highlighted blue. Those three phases are called the interphase. So the interphase, kind of is, it means in between, right? So the interphase is the in-between phase. In between what? In between each cell division. So before the cell actually divides in the mitotic phase, you can see that we spend way more time preparing for cell division. So the interphase of G1, S, and G2 are our preparation steps, right? It's kind of like, at this point, you might have heard me talking about baking a cake, baking cupcakes so many times. But you know how if you were to bake a cake, right, or make any kind of food, oftentimes you spend a lot more time preparing to make that food compared to the actual cooking process, right? If you bake sometimes, right? Not that I actually bake, but I've seen people doing it, right? There's a lot of preparation that you have to do. You have to go to the grocery store. You have to mix everything together. You have to do this and that, all kinds of things. And then you stick it into the oven, and then not too long after, it's done, right? So same thing with our cell. We got all these preparation steps, and we have to do them right before the cell actually divides, okay? So this is an overview. Make sure, hopefully, by this point, you've not only drawn all of this. If you have to pause and draw it, that's fine. But hopefully you understand, what I'm talking about, right? Is really, once again, you've heard this 1,000 times. It's more important for you to understand and remember rather than just writing everything down. It's all written down right here. It's really not as important to write it down than actually getting it, okay? Moving on. Oh, so the G1, see S phase, going on with DNA. We got G2, and then we got mitosis, the M phase, mitotic phase, okay? So this is how we do it. We go G1, S, G2, M. I know this is a little silly, it's just 4 phases, but get yourself stood up and do a big stretch. At least now you know how the sequences go, right? So repeat after me. We go G1, S, G2, M, G1, S, G2, M. What are they? Gap 1, synthesis, gap 2, mitotic phase. Good thing I don't try to sing. You wouldn't want to hear it. Okay, moving on. So now you already have a pretty good understanding of the cell cycle, right? There's a lot of preparation. There's a lot of preparation before we actually divide the cells. But let's put it into words, okay? So you can pick and choose whichever parts you think is important. This one right here, you don't have to write it down, right? Cell cycle, a series of events take place as the cell grows and divides, right? That's just a general idea. It's a definition, but there's a lot more to it than just this one sentence, right? As I've said, the interface is our preparation for cell division, and there are three parts to it. There's the G1, S, and G2. Make sure you write this down. And as you can see, once again, this takes up a lot more time than the actual cell division process, right? The M phase is our actual cell division, and there are two parts to it. We got mitosis and we got mitosis and we got cytokinesis. And here's a problem, right? Our G1, you can also see, wait, did I miss that completely? I forgot to tell you guys. Over here, there's also the G0 phase. That's the resting phase, okay? All right, going back to resting phase. So here's the thing. A cell's not always dividing, right? You might have known that neurons don't divide very frequently, which is why if someone gets a concussion, unfortunately, it takes them much longer to heal, right? Compared to, something that is visible, right? If you just break your skin, it's fine. It's not great, but it's not as big of a deal because skin cells divide really fast, but neurons don't. And one of the reasons for that is a lot of our cells actually kind of stay in the G0 phase, as in they don't need to be dividing. If they don't need to be dividing, they don't need to prepare to divide either, right? So they're neither in the interphase nor the M phase. So they're in somewhat of a resting phase, okay? So the idea is when a cell is in its G0 phase, the cell is not dividing. This part is dividing. This part is dividing. Nor is it preparing to divide. This is preparing to divide, okay? We'll talk a little bit more about G0 in a little bit. And then moving on, sorry. You can pause right here if you need to write these notes down. All right, moving on. We have just what I just said, right? Frequency of cell divisions varies depending on the cell's type, right? So we got different types of cells. And then I'm making a recording. Okay, back to it. So we got a visitor here. Here's a visitor. This is Cat. This is a cat break. Say hi. All right. I don't think he's very interested in learning about cell division, so I'm going to put him down. Actually, I'm putting him up here because he likes to stand right in front of the computer. It's nice and warm over here. Okay. So here we got frequency of the cell division varies depending on the cell type, right? For example, red blood cells. We're very often making new red blood cells, right? We do it all the time, and that's great. A lot of muscle cells. We make a lot of new muscle cells. That's how you get more muscles, right? Neurons, on the other hand, they don't divide as frequently, and that's okay. Okay, now your question for you. Which phase is longer? We got interphase or mitosis. Which one? You know which one, right? Right? It's interphase, right? From your giant pizza pie, you can see that most of the pizza is the interphase. All right. Okay. Now you don't have to write this part down. Don't write it down. We'll talk about each part of the interface right now. But just to give you again another idea, major events during the interface, we got DNA or chromosomes are replicated, nucleolus become visible. You don't need to understand what that means. Nuclear membrane remains intact. You don't need to know what that means. I don't know why this is still here. I'll get rid of it. Okay. Okay. So 2 questions for you, right? We got these checkpoints. I kind of mentioned it a little bit. Why does a cell need to regulate its growth and replication? Why can't they just be dividing all the time? Why would that be a problem? You can write down the answers in your notebook. What would happen if a cell cycle regulation mechanism is broken, right? What if we checked right here, but even though there's not enough nutrients, there's no growth factor, that's kind of a signal for cells to divide, there is DNA damage, but we wouldn't have had anyway. What would happen? If your DNA replication is not done correctly, but we wouldn't have had anyway, what would that happen, right? What would happen then? Cell size, if the cell is not big enough and you divide it into 2 halves, is that a problem? You don't know what a spindle is yet for most of you, but that's okay. But anyway, if any of those things go wrong, but you keep going anyway, is that a problem? I think the answer is pretty clear, right? You don't want to keep on going with problems. If we can fix the problem, we fix it. If we can't fix the problem, we kill the cell. Is that serious? Okay. So this is the picture right here. Once again. Okay, we're moving on. Interface. Once again, G1S and G2 are your interphase. G1 at okay, so now we're going to talk about just the G1 phase and the G1 checkpoint. You ready? Here we go. Okay, so we have G, once again, it stands for gap, right? So G1 is gap 1 phase. Gap 1 phase, because it's the first gap between each cell division. So there are several events that happen. There are way more than just 3 events, but this is all I need you to know. We got cell increase in size, right? Your cell gotta get a little bigger. We're trying to fit more things into the cell. We're actually trying to fit double the amount of things in the cell. The cell needs to get bigger, right? So the cell needs to get bigger. That's great. We need some new proteins, new organelles. Once again, if you only have one set of things and then you divide it, right? It's like if you want to eat the whole cake and then you divide it in the middle, you have to split it with your brother. That's not great. But what if someone made another cake? So you both get one full cake. Wouldn't that be great, right? That is way too much cake. I don't know how much cake you eat. That's too much cake for me. But the idea here is, in order for us to go from 1 cell to 2 cells, you have to double everything inside, right? That would only make sense. Prepare for DNA replication. So we go from G1 to the S phase, right? So before DNA replication happens, we have to prepare for it. How do we prepare for it? We check to make sure our DNA has no damage, right? So this leads us to the G1 checkpoint. We keep going, and then we stop at the G1 checkpoint. Should we go or not? This is when the cell asks itself that question. Do we keep going or do we stop right here? What do we check for? We check for the things that we were preparing for, right? Is there enough nutrient? Are there growth factors? Once again, the growth factors are, we'll talk about it, but the growth factors are signals telling cells, oh, it's time to divide, right? So for example, if you break some skin, the cells surrounding that area are really going to talk to each other and go, it's time to divide, guys. We got a problem here. Let's fix it. Our cells are really good at fixing problems. And me, on the other hand, okay, anyway. DNA damage, right? I said before our DNA replicates, we have to make sure that DNA is not damaged. We don't want all kinds of DNA problems before we make double the amount of problems, right? Okay, so what happens if we don't pass the G1 checkpoint? We will go into the G0 state, which is the resting phase. All right, moving on to the S phase and the S checkpoint. S, once again, is for DNA replication. We call it the synthesis phase. You don't need to write this down again because we've already done that. Event, DNA replication. We've already, had too much dinner. DNA replication, we've already written this down, right? S checkpoint, what do we check for? We're doing DNA replication, but just like, sometimes certain people like to copy some homework. It's okay. It happens, right? But don't do it anymore. You're in high school. Anyway, but even if you got a paper in front of you and you're copying down every single word, you still might make some errors, right? Right? The errors happen very often, actually. So same thing. With DNA replication, there could be errors. There could be parts of the DNA where we simply copy wrong within our cells. So what do? We fix it. We try and fix it, and we can't fix it, we kill the cell. DNA damage, DNA replication errors. Once again, we go from 1 DNA to 2 DNA. This right here is a sister chromatid to this right here. They are identical copies. This right here is the sister chromatid of this piece of DNA, and they're identical copies as well. Moving on, we're on to G2. We're almost done. G2 is kind of our last line of defense, right? Our M phase, G1S, G2M. M is cell division. So before we get into cell division, we basically just got to check everything one more time to make sure we're good to go, right? So what do we check for? Organelles, molecules required for cell division are produced. We check to make sure that the cell is big enough, that the DNA replication is done correctly, there's no damage to the DNA itself. Okay, so you're welcome to pause and write this down. Cool. All right. Mom said I can't do mitosis until I finish my interphase. Okay. Lastly, we have our M phase. We finally made it to the M phase. It is time for cell division, right? So cell DNA is already replicated. We're gonna go and divide one cell into two cells now. So M phase, once again, is the mitotic phase. Cell division resulting in two daughter cells. You don't have to write this down if you already know it, right? The M phase includes mitosis and cytokinesis. We've already written this down, so you actually don't have to write it down again. But if you want to, that is all right. Here's a quick review for you. 4 phases of the cell cycle. G1, S, G2, M. For gap 1 phase, synthesis phase, gap 2 phase, mitotic phase. Those are our 4 phases. The interphase includes G1, S, and G2 phase. 2 steps of the M phase are mitosis and cytokinesis. Which phase are the chromosomes replicated? That's the S phase, because synthesis, DNA replication. G0 phase is the resting phase. When does this, you should be answering this. But I'm just telling the answers, just in case. When does a cell enter the G0 phase? When a cell shouldn't be divided, or if there's an error that is not fixed yet in the G1 phase, then we'll enter the G0 phase. We'll fix it up, and then we'll go back into the cell cycle. It's kind of like it's winter, the bear is going dormant. That's kind of the G0 phase. It'll wake up, or it might not. If it doesn't wake up, we kill it. But by it, I mean just the cell. Okay, number 6. What happens during the G1 phase? Okay, we just did this. Okay, cool. So at this point, if the class already ended, you can stop right here. But if class hasn't ended, keep watching. Cell cycle regulation. I'm just going to keep going, okay? Cell cycle regulation. We got the cell cycle. We talk about these different checkpoints, but that's not the only thing, okay? There are also these things called regulatory proteins. A lot of the jobs in the cells are being done by proteins. A lot of the jobs in our body are being done by the proteins. It's really not the DNA that's doing the actual jobs. The proteins doing the actual jobs. Okay. So regulatory proteins are in charge of regulating the cell cycle. There are 2 types of it. There's the internal regulator, as in this regulatory protein comes from the cell itself. The cell can send itself a signal and go, oh, it's time for me to be replicating. I will do that. As an example, there's a regulatory protein called P53, which is a tumor suppressor gene that makes a tumor suppressor protein that suppresses tumor, right? So that is a signal to tell the cell not to divide when it's not supposed to, which is also really important, right? You got the self-control for the cells. And then there are also these external regulators, like I mentioned earlier, that if you get, certain damages to your skin, or whichever part of your body, really, the cells will send signals to each other and tell each other that it is time to divide. What are you waiting for? Those are called growth factors. Okay, so you need to know growth factors. Okay, so what happens if a cell cycle regulation fails, right? You can see from these pictures, normally your cell is supposed to be nice and flat and staying in a layer and be exactly where it's supposed to be, but that doesn't always happen. This even happens to plants sometimes. Any organisms can get this. It's really unfortunate. So what really happens, though? At the beginning, it might just be a little bit of a mutation. May or may not be good or bad or neutral, but it's only a little bit of a mutation. But if you get too much of it, it could potentially cause cancer. And the cells can keep dividing without being regulated. It doesn't matter whether another cell is telling it to start dividing or stop dividing. or not, it will just keep dividing no matter what, okay? As you can see, cancer cells can divide and spread. This is kind of to show you normal cells, when there are growth factors being given to them in a petri dish, they're supposed to divide. When there aren't growth factors, they're not supposed to divide, right? So normal cells, they all divide kind of in one layer. They're not supposed to pile on top of each other, really, oftentimes. Cancer cells, on the other hand, with or without growth factors, they will just keep growing, and they can pile on top of each other. And that's how a tumor is formed in a nutshell. It's really a lot more complicated than that. And I'm clearly not a cancer biologist, but who knows? Maybe one day. Anyway, last thing that we're going to talk about today is called apoptosis. I love this word, apoptosis. It just makes sense. The cell is kind of popping, but it's kind of a terrible kind of popping. It actually dies. But apoptosis is what we call a programmed cell death, as in this is not an accident, right? A cell is either programmed to die, right? Or as a part of the natural development of the organism, or if a cell is damaged, or let's say that you got a foreign invader kind of cell into your body, a bacteria, a certain fungi, then your immune cells can tag those cells for apoptosis as well to get rid of it. So the cell kind of break into pieces, kind of. Okay, so this is to show you in reality what is this used for, right? I said this is a part of it could be a part of the program of an organism, but how does that work, right? So for example, when we are tiny babies, before we are even tiny babies, our fingers are actually all connected. We're more like ducks. But then before we're born, the membranes in between the digits will disappear. But they don't really just disappear out of, nothing, right? It's really the cells will go through apoptosis in between the fingers so that you get actual fingers. Same idea with the tadpole to a frog. When the tail disappears, they didn't really disappear, right? The cells actually went through apoptosis, the programmed cell death, and then the cell material get recycled and reused, hopefully. Normal cell, apoptotic cell. Review. Here are some questions you should be able to answer. Then you're good to go for the quiz. These ones are not on the quiz on Friday. I don't know when you're watching this, but either way, I hope this video is helpful for you, and I hope you'll have a good day.